People don't care how much you know until they know how much you care. A tele-reminder from Teleradyo Marcelo, ang telearalan ng bawat Malulenyo.
Magandang araw, Delphilorians!
Sa taong po ito, pag-iiwan natin ang isang radyo. Mabuhay ang Pilipinas!
Makinig, manood, nasa teleradyo. Makinig, manood, nasa teleradyo. Teleradyo, Teleradyo Marcelo. Sa gitna ng mapanubok.
Good morning! Welcome to Teleradyo Marcelo, ang telearalan ng bawat Malulenyo. Good morning, grade 8 learners. This is Ma'am Jean Legasca from Junior High School Science Department. I will be your teacher broadcaster for the hour. I hope that you are now ready to acquire new knowledge. And remember, learning is fun when shared with everyone. Last episode, you learned about the stages of cell cycle and how we produce body cells or somatic cells in mitosis, and how the body produces sex cells in a meiosis process. Let us have a short review by identifying the stages of mitosis and meiosis in the following illustrations. Illustration number 1. What stage of mitosis is identified in the illustration? You have 5 seconds to answer. Okay, let us see if you got the correct answer. Yes, you're right. It is metaphase. Okay, let us proceed. Illustration number 2. What stage of mitosis is identified in the illustration? Again, you have 5 seconds to answer. Okay, let us see if you get the correct answer. Yes, it is anaphase. Let us proceed. Illustration number 3. Again, what stage of mitosis is identified in the illustration? 5 seconds. Okay, let us see your answer. Yes, you're right again. It is telophase. Let us continue. Illustration number 4. What stage of mitosis is it? 5 seconds. Okay, you are right. It is prophase. And last but not the least, illustration number 5. What stage of meiosis is identified in the illustration? 5 seconds. Yes, it is telophase 2. Very good. You are already know the different stages of mitosis and meiosis and the important events in each stage. Today, we will be discussing the significance of meiosis in maintaining the chromosome number. But before that, let us know first what is our most essential learning competency for today. Okay, our most essential learning competency is to explain the significance of meiosis in maintaining the chromosome number. Our objectives for today's lesson are: number 1, make a graphic organizer that differentiates spermatogenesis and oogenesis. Number 2, appreciate the significance of meiosis in reproduction, variation, and in maintaining the chromosome number of species. And number 3, understand the consequences when something goes wrong with meiosis. Okay, to begin our discussion on the importance of meiosis, let us know first what are chromosomes. Okay, chromosomes are thread-like structures, as illustrated at the right side, located inside the nucleus of plant and animal cells. Each chromosome is made of protein and a single molecule of DNA, or deoxyribonucleic acid, passed from parents to offspring. DNA contains the specific instructions that make its type of living creature unique. Let us watch this video so that we can understand more what are chromosomes.
We are asked to fit a thread of 1 meter in this small box. It will be easy if we simply coil the thread and place it inside. Did you know that a similar approach is taken by our cells? Let's see how. We know that the long stretch of DNA is coiled to a great extent to fit inside the tiny nucleus. This process of coiling gives rise to the chromatid structure. It's a thread-like mesh that we find in the nucleus of a metabolically active cell. The chromatids gain a typical classical structure which looks somewhat like this. It's seen during cell division. And these are nothing but the chromosomes. But tell me, do all chromosomes look exactly like this? Do all the 46 chromosomes look the same? If yes, then why do we say that there are 23 pairs of chromosomes? What helps us categorize these chromosomes as pairs? Well, all chromosomes are not exactly like one another. They come in types depending upon their structure or shapes and functions. The first basis for classification is shapes. We know this is the structure of a typical chromosome. And is this a single chromosome? That's right. The difference is that this chromosome is not duplicated, while this one has a duplicated copy attached to itself. So these are the duplicated sister chromatids which are attached to the same chromosome. Now, to understand the types, let's first understand the typical structure. Here, these two regions are called the arms of the chromosome. This arm is named as the P arm and this is known as the Q arm. So how do we recognize which is the P and which is the Q arm? Is it based on their positions? Nope, it depends on the size. The smaller arm is the P arm while the larger one is the Q arm. So this chromosome has one P and one Q arm, while this one has two P and two Q arms. Now let's focus on this chromosome structure. Can you guess what this structure is called to which the arms are attached? Well, this knot-like structure is called centromere. It's the portion of DNA that acts as a link between the two sister chromatids. But that's not its sole purpose. Its major function is the attachment of spindle fibers during the cell division process. And let me tell you one more interesting use of the centromere. It's the position of the centromere that helps us categorize the chromosomes into 4 types. The 4 types include metacentric, submetacentric, acrocentric, and telocentric chromosomes. Let's have a look at the structures of each type. Aren't these simple to understand? This one is called metacentric type. As we can see, the centromere is placed almost exactly at the center. The 2 arms seem almost of the same length. We can say that the centromere is placed somewhat in the middle of the complete structure. Now the next type is the submetacentric. Here, the centromere is placed slightly away from the center. As a result, the 2 arms of the chromosome appear unequal. The next type is the acrocentric. In this, the centromere is seen almost towards the end region. The term acro in Greek relates to peak. Thus, the name acrocentric refers to the type where the centromere is near the terminal region. Lastly, what we see is that the centromere is present at the extreme end. That is, the centromere is placed at the telomeric region. Hence the name telocentric. However, these chromosomes are not found in humans. The human karyotype lacks telocentric chromosomes. But what is karyotype, you may ask? Well, it's one way of representing all the chromosomes found in a cell. This is how chromosomes are classified on the basis of their structures or shapes, to be precise. Now let's have a look at the other type of classification. This is a much simpler type of classification. Here, the chromosomes are classified whether they are autosomes or sex chromosomes. Now what could these be? This is a representative image of a typical human karyotype. The chromosomes that pair from 1 to 22nd are called autosomes. That is, they are not the sex chromosomes. To be precise, they have got no function to deal with the sex of the individual. The last pair, however, determines the sex of the person. So if the 2 are identical, that is, both are X and X, then the individual is a female. And what if both are different? In such a case, when one chromosome is X and the other is Y, the individual is a male. This pair is the sex chromosomes. So the chromosomes which help in determining the sex of an individual are sex chromosomes, or even called allosomes. The others, which are from 1st to the 22nd pair, are not the sex chromosomes. This makes them the autosomes. Both contain genes that code for proteins which make up our body. Now that we've understood the various types of chromosomes, can you tell me what helps in determining the sex of an individual? Is it only the chromosomes? Or is it some other factor too? Tune into the next part to understand the interesting concept of sex determination in various organisms.
I hope that with this video, you understand more about chromosomes. Every species has a specific number of chromosomes. The chromosomal number is either diploid or haploid. Diploid means the nucleus of a cell contains 2 sets of homologous chromosomes. On the other hand, haploid cells contain chromosomes in a single cell, thus it is equal to half of the diploid number. Let's try to answer this one. A donkey has 62 chromosomes, and a zebra has 46 chromosomes. The owner of the donkey and the zebra mate the two and produce a zonkey. How many chromosomes does a zonkey have? Can you guess it right? Okay, 5 seconds. Okay, let us see if you got the correct answer. It is 54. Okay, let us. I have here a table with a column for the species name, a column for the number of chromosome in diploid cells, which is N, and a column of chromosome in haploid cells, which is N. Complete the following table of chromosome number in various species. Okay, you have 5 seconds to answer. Okay, let us see if you got the correct answer. Let us see. In housefly, the number of chromosome in diploid cells is 12, and the number of chromosome in haploid cells is 6. In monkey, the number of chromosome in diploid cells is 42, and the number of chromosomes in haploid cells is 21. In bat, the diploid cells is 44, and the chromosome in haploid cells is 22. In king crab, the number of chromosome in diploid cells is 208, and the number of chromosome in haploid cells is 104. In goat, the number of chromosomes in diploid cells is 60, and the number of chromosomes in haploid cells is 30. And in rice, the number of chromosomes in diploid cells is 24, and the number of chromosomes in haploid cells is 12. Okay, very good. Homologous chromosomes are pair chromosomes having the same appearance, carrying both the genes that control the same traits except for reproductive or sex cells. On the other hand, non-homologous chromosomes are chromosomes that do not belong to the same pair. Generally, the shape of the chromosome, that is the length of the arm and the position of the centromere, is different, like this one. Okay, let us try this one. I have here figure number 1 and figure number 2. In figure 1, I have here sets of chromosomes from A to E. Figure 2, haploid chromosomes and diploid chromosomes. Question number 1. Which pairs or pair of chromosomes or chromosomes is our homologous chromosome? From figure 1. Okay, let us see. Yes, you're right. Uh, Chromosome A, chromosome C, and chromosome E are homologous chromosome. On the other hand, chromosome B and chromosome D are non-homologous chromosome. Okay, figure 2. What is the haploid chromosomal number? Yes, that is 3. And what about the diploid number? Yes, that is 6. Humans have 46 number of chromosomes. 23 of these came from the father's sperm cells, while the other 23 from the mother's egg cell. During fertilization or during sexual reproduction, the haploid sperm cell unites with the haploid egg cell to form a fertilized egg or zygote, which is diploid. The zygote will grow and develop into a multicellular adult by mitosis. What makes it possible to produce haploid cells? Do you know the answer? It is through meiosis. Because meiosis is a special type of cell division of germ cells in sexually reproducing organisms used to produce the gametes, such as sperm and cells. It involves two rounds of division that ultimately result in 4 cells with only one copy of each chromosomes. Let us know the relationship between meiosis and sex cells. Gametogenesis is a process where sex cells undergo meiosis to form sperm cells in male and egg cells in female. Spermatogenesis is the production of sperm by the process of meiosis. It begins actually at the age of puberty, more or less from 13 to 16 years of age, and continues until old age. On the other hand, oogenesis is the process of producing egg cells by means of meiosis. The production of oogonia by means of mitosis begins during the prenatal stage in the development of a human female. The produced oogonia will be released in each lifetime. Let us watch this video so that you can well understand what is spermatogenesis and oogenesis.
In this video, we will study about the process of spermatogenesis. Spermatogenesis is the production of mature male gametes which are known as sperms. About 200 to 300 million sperms are produced daily in the male gonads known as the testes. These immature sperms also undergo a process of maturation in the epididymis to form the mature sperms. For a better understanding of this topic, make sure to watch our video on the male reproductive system. As we discuss the anatomy of the testes in the male reproductive system, the testes contains numerous pipe-like structures known as seminiferous tubules. The seminiferous tubules have a central lumen and outer wall. The wall of the seminiferous tubule is the main site for the production of sperms. Now let's look at a closer view of the cross-section of the wall of seminiferous tubule. As we can see, the wall of the seminiferous tubule contains numerous types of cells, and one of them are the primary germ cells. The primary germ cells transform through various stages of development and finally form immature sperms known as spermatids. Now let's talk about the process of spermatogenesis in a bit detail. The process of spermatogenesis is divided into two phases. The first is the spermatocytogenesis, in which one germ cell divides into 4 precursor gametes known as spermatids. These spermatids are immature sperms. The second phase is the spermiogenesis, in which no further cell division occurs, but the immature sperms go through the process of maturation and differentiation to form mature sperms. So now let's talk about the series of cell division that occurs in the first phase of spermatogenesis. We start with the primary germ cells known as the primary spermatogonium type A, which are found in the wall of the seminiferous tubules. The main job of primary spermatogonium type A is to divide into 2 types of cells. The first is the type B primary spermatogonium. The type B spermatogonium auto-reproduce and maintain the number of the primary spermatogonium type A. The first division in the series of spermatogenesis is a mitosis, which gives rise to an intermediate stage known as primary spermatocyte. The primary spermatocyte are diploid cells maintaining the number of chromosomes. In the second division, the primary spermatocyte go through the first phase of the meiosis and divide their chromosomal material into half to give rise to secondary spermatocytes, which are haploid cells. In the last stage, the secondary spermatocytes again go through the process of meiosis II and maintain their chromosomal number. They give rise to 4 immature sperms known as spermatids. Looking at the wall of the seminiferous tubule in a bit detail, the primary spermatogonium are located in the basal layer of the wall of seminiferous tubule, and in the center you can see the various divisions from primary spermatocyte till the spermatids. During all these cell divisions, the Sertoli cells give support and nutrition to the developing gametes. So now we know that one germ cell gives rise to 4 spermatids. But spermatids are immature sperm cells incapable of fertilization, and they contain numerous cell bodies like Golgi body, centrioles, and mitochondria. The spermatids undergo through the process of spermiogenesis, which is second phase of spermatogenesis, to form mature sperms. The process of spermiogenesis is basically divided into 4 phases, which converts an immature sperm into a mature sperm. The first is the Golgi phase, where the Golgi body present in the spermatid forms the enzymes of the acrosome. The second is the acrosomal phase, where the formed acrosome condenses around the nucleus to form the cap. The third is the tail phase, where the centriole present in the spermatid forms the tail of the sperm. And the last is the maturation phase, where the spermatid loses the excess cytoplasm and forms the mature sperm. But the mature sperms are still non-motile. As we know, the process of spermatogenesis runs in the wall of the seminiferous tubule. The mature sperms are then released in the lumen of the seminiferous tubule through a process called spermiation. The sperms then spend a few days in the epididymis and gain motility. The motile sperms are thus capable of fertilization. So this was all about the process of spermatogenesis. I hope you found this video helpful. Thank you so much for watching this video. Like our channel on YouTube for all the upcoming videos, and make sure to like our channel on Facebook for flashcards and upcoming notifications.
Hi guys, welcome back to Embryology with Anatob. In our last lesson, we discussed about gametogenesis. Well, in today's lesson, we will be discussing about oogenesis. Oogenesis is a process whereby oogonia differentiate into mature oocytes. Maturation of oocytes can be divided in 2 stages: maturation before birth and maturation at puberty. Let's begin with maturation before birth. Once primordial germ cells arrive in the gonad of a genetic female, they differentiate into oogonia. Once oogonia are formed, they undergo a number of mitotic divisions, and by the end of the 3rd month, the oogonia become arranged in clusters surrounded by a layer of flat epithelial cells known as follicular cells. These follicular cells originate from the surface epithelium covering the ovary. During the next few months, oogonia increase rapidly in number and enlarges to form primary oocytes, and by the 5th month of prenatal development, the total number of germ cells in the ovary reaches its maximum, which is estimated at about 7 million. By the 7th month, cell death begins to occur, and majority of oogonia will degenerate, leaving about 2 million primary oocytes. Now the remaining 2 million primary oocytes begin to undergo meiosis. A primary oocyte combined with its surrounding follicular cells is known as primordial follicle. About the period when the primary oocyte enters the prophase stage of meiotic division, follicular cells surrounding the primary oocytes begin to release a small peptide called oocyte maturation inhibitor, also known as the OMI. Now the OMI arrests the meiotic division at the diplotin stage of prophase so that the primary oocytes do not finish their first meiotic division before puberty is reached. Now let's talk about maturation at puberty. The total number of primary oocytes at birth is estimated to vary from 700,000 to 2 million. During childhood, most oocytes become atretic. Only approximately 40,000 will be present by the beginning of puberty, and fewer than 500 will be ovulated. Once puberty begins, 15 to 20 primary oocytes begin to mature each month, although only one of these reaches full maturation to become an oocyte. Maturation of primary oocytes undergo 3 stages: the pre-antral stage, the antral stage, and the pre-ovulatory stage. In the pre-antral stage, surrounding follicular cells change from flat epithelial cells to form a stratified cuboidal epithelium called granulosa cells, which will go ahead to form the zona pellucida around the primary oocyte. Together, they form the primary follicle. Surrounding connective tissue cells also differentiate into theca folliculi, which are responsive to luteinizing hormone. In the antral stage, central fluid-filled space called the antrum forms between granulosa cells. The follicles are now called secondary follicles. Granulosa cells surrounding the oocyte remain intact and form the cumulus othorus. Now to the pre-ovulatory stage. When the secondary follicle is mature, a surge in the luteinizing hormone induces completion of meiosis 1. 2 haploid cells are formed within the follicle, but they are unequal in size. One of the daughter cells receives far less cytoplasm than the other and forms the first polar body, which will not go ahead to form an ovum. The other haploid cell is known as a secondary oocyte. Both daughter cells then undergo meiosis 2. The first polar body will replicate to give 2 polar bodies, while the secondary oocyte is arrested at the metaphase stage of meiosis 2, about 3 hours prior to ovulation. Now, if a sperm penetrates the secondary oocyte, the second meiotic division is completed and most cytoplasm is again retained by one cell, the fertilized oocyte, and the other, the second polar body. That will be all for now, guys. Now, if you like this video, please give us a thumbs up. Don't forget to subscribe, share, and leave us a comment. Thank you. In our next lesson, we will be discussing about spermatogenesis.
Okay, let us try to summarize the process of spermatogenesis and oogenesis. In spermatogenesis, at puberty, these one primary spermatocytes divide by means of meiosis 1 to produce 2 haploid secondary spermatocytes. The secondary spermatocytes undergo meiosis 2, and each cell produces spermatids that are haploid. This results in a total of 4 spermatids that will mature into form sperm cells. In oogenesis, in meiosis 1, one oogonium transformed into one primary oocyte. As meiosis 1 finally complete, a primary oocyte divides into 2 haploid cells of unequal sizes. The large cell is called the secondary oocyte, and the small one is called the first polar body. Both cells enter meiosis 2, and the division process occurs again. The secondary oocyte divides again into 2 unequal sizes: a haploid ovum or egg cell and a second polar body. The first polar body also divided into 2 polar bodies. All the polar bodies will soon degenerate. The entire process of oogenesis results in only one haploid ovum. this figure. Write the chromosomal number of the cell in its stage of meiosis, whether it is diploid or anhaploid, from primary oocyte and primary spermatocyte. Then you have there the secondary oocyte, the secondary spermatocyte, then you have there the mature ovum, the polar bodies, and the spermatids. Write the chromosomal number of the cell in its stage of meiosis, whether it is a diploid, which is 2n, or a haploid, which is n. Okay, you have 5 seconds to answer. Let us see if you got it right. Yes, you are right. Primary oocyte is diploid 2n. Primary spermatocyte also diploid 2n. The secondary oocyte as well as the secondary spermatocytes are haploid, which is n. The mature ovum, the polar bodies, and the spermatids are all haploid. Okay, after going through with chromosomes, the process of spermatogenesis and oogenesis, let us now study the importance of meiosis. Why is meiosis important? Meiosis is important to sexual reproduction. It forms the correct haploid number of chromosomes. This maintains the correct diploid number of chromosomes in organisms when sex cells joined. The picture shows a mother with a diploid chromosomes and a father with a diploid chromosomes too. After meiosis, the diploid chromosomes became haploid. So, when the egg cell and the sperm cell unite, a diploid organism is produced to maintain the chromosome number of an organism. Meiosis is also important to genetic variation. Genetic variation. Genetic variation exists among humans. You can look at your friends to see genetic variation. Some people have blue eyes, others have brown eyes, and some people have blonde hair, others have red hair, and others have black hair. Genetic variation occurs in all organisms that reproduce sexually. Again, what are the advantages of sexual reproduction in genetic variation? Because of genetic variation, individuals within a population have a slight differences. These differences might be an advantage if the environment changes. Some individuals might have traits that make them able to survive harsh conditions. For example, some plants within a population might be able to survive long periods of dry weather. Sometimes the traits might help keep an organism from getting infected by a disease. To answer this one, read carefully the following statements. Tick the yay column if it describes the importance of meiosis, and nay if it is not. I have here at least 5 situations. Number 1, if sex cells won't divide through meiosis, the chromosomal number will continue to increase. Is it a yay or a nay? Number 2, meiosis ensures that all the chromosome number of organisms changes from generation to generation. Number 3, meiosis safeguards that its daughter cell receives a copy of its kind of chromosomes. Thus, its daughter cell would receive its kind of genes. Number 4, the crossing over and independent assortment bring about remarkable variations in the characteristics of the organisms. And number 5, variations in characteristics among organisms is not important in adapting to the changing environment. Okay, all you have to do is just to tick The yay column if it describes the importance of meiosis, and nay if it is not. You have 5 seconds to answer. Okay, let us see if you get the correct answer. Okay. Did you get it all right? Okay, so you have there. Number 1, if sex cells won't divide through meiosis, the chromosomal number will continue to increase, and it is a yay. It means that it describes the importance of meiosis. And number 2, meiosis ensures that all the chromosome number of organisms changes from generation to generation. And it is a nay. It is not. Number 3, meiosis safeguards that its daughter cell receives a copy of its kind of chromosomes. Thus, its daughter cell would receive its kind of genes. And it is also a yay. Number 4, the crossing over and independent assortment bring about remarkable variations in the characteristics of the organism. It is also a yay. Describes the importance of meiosis. And number 5, variations in characteristics among organisms is not important in adapting to the changing environment. It is not. Okay, did you get it all right? I hope so. Let us proceed. What if something goes wrong with meiosis? What if? Most cells in the human body contain 46 chromosomes, as mentioned, one pair of 23 chromosomes from the mother, and one pair of 23 chromosomes from the father. When chromosomes do not divide correctly during meiosis, gametes will end up with too many or too few chromosomes. If a zygote has too many or too few chromosomes, it will not develop properly, and perhaps it will die. Variants also occur when a segments of a chromosomes are reversed, misplaced, or missing. Let us have some examples of this. Okay, let us see some examples of errors. One is what we call the Patau syndrome, which is trisomy 13, which result from 3 copies of chromosome 13. Problems include heart defects, weak muscle tone, intellectual disabilities, brain disorders, and cleft lip. Babies with trisomy 13 have a high mortality rate within the first year of life. Another we call the Down syndrome is a condition known as trisomy 21. Down syndrome is due to an extra copy of chromosome 21. Children born with Down syndrome experience developmental, cognitive, and intellectual delays ranging from mild to severe. Another one is what we call the Klinefelter syndrome. Occurs when males have an extra X chromosome. So that is XXY. The condition can inhibit growth of genitalia, hormone production, and fertility. The other one is what we call the Jacob syndrome. Males born with an extra Y chromosome. So that is XYY. Okay, the symptoms include language difficulties, problems with sitting and walking, and behavioral emotional issues. People with an extra Y chromosome may also have mild autism and weak muscle tone. Okay, I hope that you learned something, and let us test your learning. It is quiz time. Okay, number 1. Start. Number 1. What is the type of cell division that produces gametes and prepares the organism for fertilization during sexual reproduction? Letter A, cytokinesis. Letter B, meiosis. Letter C, mitosis. And letter D, synapsis. Okay, you have 5 seconds to answer. Okay, let us see if you get the correct answer. The correct answer is letter B. Okay, let us proceed. Number 2. Which of the following is false about oogenesis? Letter A, oogenesis begins during fetal development. Letter B, oogenesis produces haploid cell unidentical to the parent cell. Letter C, puberty is the female trigger's primary oocyte to continue their development. Letter D, oogenesis produces one primary oocyte, two secondary oocytes, and four ova from one oogonium. False about oogenesis. Okay, 5 seconds. Okay, let us see if you got it right. The correct answer is letter D. Oogenesis produces one primary oocyte, two secondary oocyte, and four ova from one oogonium. It is false. Okay. Okay, number 3. If there are two oogonia, how many ova or egg cell will be produced after meiosis? Letter A, 1 egg cell. Letter B, 2 egg cells. Letter C, 3 egg cells. And letter D, 4 egg cells. Go. Okay, let us see if you got the correct answer. The correct answer is letter B, 2 egg cells. Let us proceed. Okay, question number 4. Blank is a condition known as trisomy 21. Letter A, is it a Patau syndrome? Letter B, a Down syndrome. Letter C, Jacob syndrome. And letter D, Klinefelter syndrome. Go. Okay, the correct answer is letter B, Down syndrome. That is trisomy 21. Okay, let us proceed. Question number 5. Blank are thread-like structures inside the nucleus of plant and animal cells. Letter A, is it egg cells? Letter B, sperm cells. Letter C, somatic cells. And letter D, chromosomes. 5 seconds. Okay, let us see if you got it right. The correct answer is letter D, chromosomes. Very good. Okay, your assignment for the next episode is to study the Mendelian genetics. That's it for today. Thank you for joining today's class. This is Ma'am Jean Legasca from Junior High School Science Department. Thank you and good day.
at your service. Kaya naman, ang feedback mo, suggestions nyo ay mahalaga sa amin to improve our programs on air and online. Para alam ni teacher yung strengths as well as the things in your mind na mas makatutulong sa mas effective na pagkatuto sa teleradyo. Please take time to send your feedbacks via Teleradyo Mu, which you can access by scanning the QR code on the right or typing the URL provided here, bit.ly/3ORVAY9. One more, this time you follow. bit.ly/3ORVAY9. Kaya send your hashtag TBH o hashtag RT dito sa Teleradyo Mu kung saan ang feedback mo, suggestions nyo ay mahalaga sa teleradyo.
Makinig, manood, nasa teleradyo. Makinig, manood, nasa teleradyo. Teleradyo, Teleradyo Marcelo. Sa gitna ng mapanubok na panahon, magkahatid sa kaakibat